Imagine biting into a crisp, fresh cabbage leaf, um, completely unaware that its cells have actually been engineered to produce the venom of a predatory animal.
Yeah, it definitely sounds like a plot from a sci-fi thriller or something.
Right,
Yeah.
but it is a very real reflection of where biological science stands today. And, well, welcome to another deep dive. Today we're looking at something that affects all of us. For thousands of years, our ability to shape nature was basically limited to picking which seeds to plant or, which animals to breed.
Which is a very slow observational process.
Exactly, it took forever. But today, incredible new technologies allow scientists to just bypass the weight and rewrite the actual source code of life itself.
We really have crossed a threshold from merely observing inheritance to actively engineering it.
And that brings us to the core mission for this deep dive. We are exploring a fascinating and incredibly weighty question for you listening today. How have humans changed the inheritance of traits? And honestly, what responsibilities actually come with wielding that kind of power?
To understand the power to change inheritance, we first have to understand the raw material we are working with, right? Which is biological variation.
The baseline of all life.
Exactly.
Yeah.
If you look at any population in nature, variation actually exists. You see it in a group of giraffes with slightly different neck lengths or garter snakes with varying scale patterns.
Let's break down where that variation actually comes from, though. Because things don't just randomly change for no reason.
Well, sometimes they kind of do.
Wait, really?
Yeah, in a sense. A significant source of variation comes from random mutations. Every time a cell divides, it has to copy its DNA. And sometimes it just makes a typo.
Oh, okay.
Most of the time, these typos, or mutations, don't do anything at all. But occasionally, they alter a protein just enough to introduce a slightly different physical trait.
And then reproduction gets involved.
Right. You combine those mutations with sexual reproduction, which constantly shuffles the genetic deck, passing half the genetic material from each parent to the offspring. The result is a population filled with uniquely diverse traits.
So you've got this constantly shuffling deck of traits. But there is a massive misconception we really need to clear up right away.
The vending machine myth.
Yes. We hear headlines all the time claiming scientists found, the gene for being good at math or the gene for height. It trains us to think inheritance is a simple transaction, like you press the tall button, you get a tall person.
And biology heavily resists that kind of simplicity.
Yeah.
Desired traits are almost never controlled by just one gene.
Right.
Biological outcomes are driven by probability, multiple interacting causes, and just profound environmental influences.
Think about trying to grow one of those championship giant pumpkins you see at state fairs.
Yeah.
The ones that weigh as much as a small car.
Those are wild.
They are.
Yeah.
But you can't just buy a seed with giant pumpkin genetics, throw it on a dry patch of concrete, and expect a 1,000-pound winner.
Definitely not.
Growing a record breaker requires the inherited genetic potential working in tandem with the perfect environmental conditions. So, exact soil chemistry, precise watering, optimal sunlight. It is always a complex dance between inherited information and the environment.
And humanity figured out how to manipulate that dance long before anyone knew what DNA or genes even were.
Which is crazy to think about.
It is. But recognizing how we did that requires making a really hard distinction between natural selection and artificial selection. So in natural selection, the environment applies the pressure.
Like the garter snakes and newts from our sources.
Right.
There's a population of newts that slowly evolved to become highly poisonous over many generations, simply because the predators in their environment were eating the less poisonous ones.
The environment essentially dictated who survived long enough to pass on their genes.
But in artificial selection, which we usually call selective breeding, humans become the environment.
Exactly. We decide which traits are desirable, and we choose which organisms get to be the parents of the next generation.
We basically take the wheel, and I was stunned by how this played out historically with agriculture. Take maize or corn. The desired trait for indigenous farmers in the Americas was a plant that yielded more food, was easier to harvest, and, tasted better.
Sure.
But their starting material wasn't anything resembling a modern corn cob. It was this wild, bushy weed called teosinte.
Teosinte is fascinating because its seeds are encased in incredibly hard shells.
Yeah.
I mean, you could break a tooth trying to eat it.
Oh, wow.
Yeah, and the ears were only an inch long.
So the farmers used selective breeding. Year after year, they scoured their fields for the rare mutant teosinte plant that had a slightly softer seed case, or maybe just a few extra kernels.
And they saved those specific seeds and planted them the next season.
Just over and over.
Right. By repeating that precise selection process across hundreds of generations, those indigenous farmers fundamentally shifted the gene pool. They made the selected traits the dominant traits of the entire population.
And today, maize is so heavily modified from its ancestor that it literally cannot survive or reproduce without human intervention.
Which is a huge shift.
And the cabbage family is an even wilder example. I mean, I've eaten cabbage, broccoli, kale, Brussels sprouts, and cauliflower my entire life. Finding out they all originate from the exact same plant, the wild Brassica oleracea, or wild mustard, that completely blew my mind.
It is a literal masterclass in how selective breeding isolates specific traits.
Yeah, you want to eat leaves? Keep breeding the plants that happen to grow slightly larger leaves, and eventually you get kale.
Right. Or if you prefer the terminal leaf buds, you breed for those and you get cabbage.
If you breed for the lateral leaf buds along the stem, Brussels sprouts. If you select for the flower clusters, you get broccoli.
The intended benefit there is massive. Farmers get specialized, easy to grow crops, and we get a diverse, reliable food supply.
And we apply this exact same artificial pressure to animals, too. The evolution of livestock and working animals is driven entirely by human preference.
Absolutely.
Let's talk about the Alaskan sled dogs, actually, because this completely challenged my understanding of how animal breeding works. You look at a championship mushers team running 1,000 miles through the frozen wilderness. You'd expect those dogs to look like clones of each other.
Like a standard breed.
Right. But the sources point out that one dog might be super fluffy, like a Siberian Husky, another is short-haired and sleek like a pointer, and another is built tall and lean like a hound.
Yet, if you run a genetic test on them, the results classify them as the exact same breed.
The Alaskan sled dog. Wait, how can a genetic test say two completely different-looking animals are the same breed?
Well, this forces us to define what we actually value when we breed. With modern recognized dog breeds, like a Dalmatian or a Pug, the desired trait is almost exclusively visual uniformity. They have to match a physical standard.
Oh, I see.
But for mushers, the desired trait is purely functional. It is elite athletic performance.
Form entirely follows function. They don't care about coat color. They care about speed, metabolic efficiency, cold tolerance, and just the psychological drive to pull.
Right. And to achieve that, mushers rely heavily on hybridization and advanced animal husbandry. They crossbred Siberian Huskies for their extreme cold tolerance, German short-haired pointers for their explosive speed, and even Anatolian Shepherds for their unwavering work ethic.
Anatolian Shepherds are those massive flock-guarding dogs, right?
Yeah, they are huge.
So they reshaped the inheritance of these dogs to concentrate physiological traits rather than cosmetic ones. But they didn't just let dogs run loose and hope for the best.
No, livestock and dog breeders use powerful methods to speed this up. Things like artificial insemination and embryo selection are standard practice now.
Meaning breeders can evaluate the genetic potential of an animal and distribute those genes globally without the animals ever actually meeting. The sled dog example also highlights a massive risk, though. The mushers are actively mixing breeds outcrossing, right, which maintains a wide, healthy gene pool. Compare that to a bulldog. When breeders only select for one single visual trait, like a pushed-in face, the organism itself ends up paying the price.
Because highly restricted breeding focused on a single trait drastically shrinks the gene pool.
Right.
That lack of genetic diversity means non-adaptive or even harmful mutations get trapped in the population. You end up with dogs that can barely breathe or reproduce naturally. The visual aesthetic benefits humans, sure, but the cost is carried entirely by the animal's health.
So selective breeding has completely shaped our world, from the grocery store aisle to our pets. But there is a hard biological limit here. A musher can mix dog breeds for endurance, but they can't breed a dog to have gills.
No, because the genes for gills simply do not exist in the canine gene pool.
Right. And furthermore, breeding takes generations of waiting. So what happens when we want to bypass that speed limit or introduce a trait that has never existed in that species at all?
That limitation is exactly what drove the transition to modern genetic technologies. We moved from selecting whole organisms based on their outward traits to directly manipulating their internal molecular blueprints.
Which brings us back to the venomous cabbage I mentioned at the start. The desired trait there is pest and disease resistance. Insects decimate crops, and farmers need plants that can defend themselves.
The traditional method would be trying to crossbreed cabbages with tougher wild relatives and just hoping a resistant strain emerges after maybe 10 years of trials.
But the method used here is genetic modification. Scientists aren't breeding anything. They are directly transferring genetic material.
And the mechanism that makes this possible is the universality of DNA. Whether you are a cabbage, a human, or a venomous scorpion, your DNA is written in the exact same chemical language.
So scientists can isolate the specific gene in a predatory insect that produces a toxic protein and physically insert that gene into the genome of a cabbage.
Exactly. The cabbage cells read that foreign instruction manual and just start manufacturing the pesticide themselves.
The intended benefits are pretty compelling. We are looking at significantly higher yields to feed a growing population and potentially a massive reduction in the need to spray broad-spectrum chemical pesticides across the environment.
True. But pouring through these sources, the unintended consequences loom really large.
Yeah, they do.
If a plant is engineered to produce a novel toxin, how does that impact the broader ecological web? Like, does it harm beneficial pollinators like bees or butterflies?
Right.
You also have to consider gene flow. What happens if the pollen from this modified cabbage drifts on the wind and fertilizes a wild, weedy relative?
Oh, wow. You could inadvertently engineer a population of superweeds that are completely resistant to pests.
Exactly. Genetic modification allows us to solve isolated agricultural problems, but ecosystems are complex networks. Changing one variable inevitably ripples through the rest.
That's agriculture. But the stakes feel even higher when we look at human medicine, specifically gene therapy. Our sources focus on hemophilia, which is a genetic disorder where a patient's blood lacks a functional protein called clotting factor VIII. A simple cut can become a life-threatening bleeding event.
So the desired trait we want to introduce there is the cellular ability to manufacture that missing clotting protein.
And the method being used to tackle this is gene editing, most notably CRISPR-Cas9. And the origin of CRISPR is just fascinating. It wasn't invented from scratch, was it?
No, it is actually an adapted bacterial defense system. Bacteria used it for millions of years to slice up attacking viruses.
And scientists just reprogrammed it to act as microscopic molecular scissors.
The scissors analogy is helpful, but the targeting mechanism is the real breakthrough here. CRISPR uses a piece of guide RNA that acts almost like the Strel F search function on your computer.
Oh, that makes sense.
Yeah, it scans through the billions of letters of DNA code inside a patient's cell until it finds the exact mutated sequence causing the hemophilia. Once it finds a perfect match, the Cas9 protein acts like the backspace key and cuts the DNA strand right at that spot.
Wait, but if it just cuts the DNA, then what? Doesn't severing a DNA strand completely kill the cell? How does breaking it fix the hemophilia?
That's the brilliant part. A broken DNA strand triggers a massive panic response in the cell. The cell's natural repair mechanisms rush to stitch the broken ends back together.
Okay.
Gene therapy hijacks that repair process. Scientists provide a template of the healthy, functional gene. So as the cell frantically tries to repair the cut made by CRISPR, it uses that healthy template as a guide.
Essentially pasting the correct instructions into the genome.
Exactly.
The cell is tricked into curing itself. That is just staggering. But biology is notoriously messy. What happens when those molecular scissors miss their target?
That is the defining risk of gene editing, which is known as an off-target mutation. The guide RNA is highly precise, sure, but in a genome of 3 billion letters, similar sequences exist.
Right.
If CRISPR accidentally cuts a healthy gene, particularly a gene responsible for regulating cell division, the results can be catastrophic.
You're talking about runaway cell growth.
Yes, unregulated cell division is the definition of cancer. The intended benefit is curing a debilitating disease at its root. But the risk is introducing an entirely new, potentially fatal condition.
The patient carries the ultimate cost.
Which is exactly why clinical trials for these therapies demand such extreme caution.
It also requires us, as a society, to be highly critical consumers of this information. The leap from we successfully edited a cell in a petri dish to designer babies are taking over tomorrow happens incredibly fast in the media.
Oh, absolutely. We are bombarded with sensationalized headlines about genetic technologies daily. Navigating that requires a very specific set of critical evaluation skills.
I see this constantly on social media. A headline will scream, "Scientists create genetically modified superhumans." It's terrifying until you actually evaluate the source.
Right. When you encounter claims like that, the very first question must be, who created this source? Is it a peer-reviewed scientific journal or a biotech startup aggressively seeking venture capital?
I always look for the underlying data. Like, are they just making a broad claim or are they presenting actual evidence supported by research? Often, these viral articles link back to a study that only involved mice, but the headline implies it's happening in human hospitals right now.
You also have to ask what the source might be selling or opposing.
Mm.
Many organizations have financial or political incentives to either promote genetic modification as a flawless miracle or oppose it as a guaranteed apocalypse.
And what information is missing? Usually the caveats are missing. The article highlights the successful gene edit but leaves out the off-target cancer risks, or the fact that the trait they are talking about is actually controlled by 50 different genes, not just the one they edited.
Checking if other credible sources agree is your best defense there. Scientific consensus rarely rests on a single isolated breakthrough.
That critical lens is just so essential because we are moving rapidly from scientific capability into profound ethical and civic territory.
We really are.
The sources present several massive dilemmas, and we need to explore them impartially for you listening. The most glaring question is, who gets to decide which traits are desirable?
Historically, farmers and breeders made those decisions based on utility. But as we map the human genome, the line between curing a disease and enhancing a trait becomes incredibly blurry. Who defines what constitutes a genetic defect versus just natural human variation?
There is also the issue of the organisms themselves. When we engineer a cow to produce twice as much milk, does that modification benefit the cow, or does it only benefit human economics while putting immense physical strain on the animal?
We also face the threat to genetic diversity we touched on earlier. If we find the perfect genetic sequence for a high-yield wheat crop and plant it globally, we create a massive monoculture.
Millions of acres of the exact same cloned plant.
And that makes our entire global food system incredibly vulnerable. If a novel disease emerges that targets that specific genetic line, the entire global crop fails simultaneously because we engineered out the natural variation that might have resisted the disease.
Then there is the deeply uncomfortable question of access. Gene therapy isn't like picking up an antibiotic at the pharmacy. We are talking about current treatments for conditions like sickle cell or spinal muscular atrophy that cost upwards of $2 to $3 million per patient.
The economic barrier is just staggering.
Are we engineering a future with a literal sociogenetic divide? If only the ultra-wealthy can afford to edit disease out of their family line, economic inequality will literally be baked into human DNA.
Navigating these issues requires communities to rigorously weigh the benefits of curing devastating diseases and securing the food supply against the risks of ecological collapse, animal suffering, and unprecedented inequality.
How do we even begin to weigh that?
I think by recognizing the fundamental difference between describing scientific consequences and deciding what society should do. Science can sequence a genome. Science can tell us the statistical probability of an off-target mutation. Science tells us what we can do.
But science doesn't have a moral compass.
Precisely. The scientific method cannot dictate our cultural values, our laws, or our ethics. Science provides the tool. Society must choose how or if to swing it.
We've covered vast territory today. We started by looking at ancient indigenous farmers, patiently coaxing slightly softer seeds out of wild teosinte grass over thousands of years to create corn.
Right.
We examined mushers hybridizing distinct dog breeds to forge elite endurance athletes. And we plunged into the microscopic world of CRISPR, where molecular scissors search and replace individual letters of human DNA to stop diseases in their tracks.
The through line is human ambition. Our desire to shape the living world to meet our needs is ancient.
The fundamental drive hasn't changed, but the technologies absolutely have. And because we now possess the power to directly edit the blueprints of life, the responsibility to examine the evidence, question the consequences, and define our values has never been greater.
We are no longer just passengers in the evolutionary process.
We hold the pen, and that brings our deep dive to a close. But I want to leave you with one final, slightly provocative thought to turn over in your mind. For our entire evolutionary history, humanity has survived by adapting ourselves to the pressures of our environment. But if these genetic technologies eventually allow us to completely and safely control inheritance, at what point do we stop adapting to the world and start demanding that the world adapt to our engineered genetics?